microscope and identifying rare cells in blood using deformability cytometry. The projects are funded by the Department of Biotechnology, Government of India, the Bill and Melinda Gates Foundation, the Tata Trust, and the Vadhani Research Center for Bioengineering. Her area of research include microfluidic devices for healthcare applications and biological physics. We welcome you, ma'am. Over to you, ma'am.
Thank you. Can I share my screen?
Yes, ma'am.
Okay, let me just see this. Okay, so is it visible now, the slides in their full form? Because I can't see the Google window anymore.
Yes, ma'am.
Okay, excellent. Thank you. So thank you for the warm welcome, and it's a pleasure to be here. Today, I'm going to talk about what we work on, which is we develop something called microfluidic devices, and we use them for healthcare applications. Okay. So I'll basically start giving an introduction to microfluidics, and then I'm planning to take you through two of our old projects where we have used these devices for different healthcare applications. Okay? So since this is virtual, I have to give you a virtual quick tour of IIT Bombay. So this is where we are situated next to Powai Lake. This is the main building of the campus, and ours is the Bioscience and Bioengineering department, where we have some people who are trained as biologists and some people who are trained as engineers working side by side. So you can go to our website and you can see, what we do. Okay. So my research group works on different kind of applications of this technology called microfluidics. And today I'm going to pick two of those. So I'm going to talk about something that we did for oral health. And then we are also going to talk about this project which separates different blood cells. Okay, but in addition to that, as we've worked a bit on tuberculosis, we work on ear infections. So we are working a lot on something called sickle cell disease and as well as malaria. Okay, so all of these use microfluidics to some extent. If anybody is interested in the rest of these projects, please get in touch and I'll be happy to answer those. Okay. So what is microfluidics? You might have heard this term before. It deals with liquids flowing through these tiny channels, which are anywhere between, say, a micron to several hundreds of microns in dimension. When I say dimension, it means either of length, width, or height. Any one dimension, if it is in this in this length scale, then we usually call these channels microfluidic. Typically, the lengths are of the order of millimeters or even longer, but width and height is typically in the order of microns. Okay, so that's important to remember. And this field came into being somewhere in the late 90s. So there used to be a field called MEMS, or microelectromechanical systems, which again came from microelectronics. But fluid flow in these devices started, these tiny devices started around late 90s. And if you look at the number of publications, and this is sort of an incomplete list, and you will find that they keep kind of steadily keep going up, which means that people have found some use of this technology. And the biggest use of this technology has come in the area of biology. Okay. There is, in fact, a journal, more than one journal actually, that are devoted to microfluidics. So one of them is Lab on a Chip. Lab on a Chip is also a broad name of the area that I work on, and I'll tell you in a bit what that means. And this is sort of a closer look into one of the microfluidic devices. There are all these are reaction chambers. They're individually addressable. This sort of reminds you perhaps of an integrated circuit, right? And this is how actually microfluidics is also supposed to work, but for fluidics. Okay, so miniaturization. So what happens, as I just told you, when fluid flows in the micron-sized channels, the volume that you are dealing with becomes very, very small. So you are essentially dealing with nanoliters and picoliters volume of fluid. Okay, so this is not even microliters, which itself is small. Okay, so when the dimension of the channel is in micron, you can work out that the volumes become in nanoliter and picoliters. Okay, and the physics of fluid flow actually changes as you keep changing these dimensions, right? So when you have, this is sort of a cartoon of a channel, and if you have this dimension, let's say it's the width of the channel, if you have a large channel, like the pipes carrying water in your building, right, in the plumbing, if it's in the order of millimeter or larger, you can have what is called turbulent flow. Whereas if you reduce it further to micron dimension, you get something as called as laminar flow, which means that basically there are, let's say there are three liquids actually are flowing side by side in this channel. There is this red one, there is the blue one, and there is something like water in the middle. And if you, and I will play a video and you will see that they can flow side by side without mixing because of this phenomenon called laminar flow. Okay. There is something called a Reynolds number. If you don't know what it is, don't worry about it. But Reynolds number essentially decides whether the flow is going to be turbulent or laminar. And for this number to be around 2000 is where this kind of turbulent features may come. But in microfluidics, forget 2000, we are dealing with numbers that are less than 1. So definitely the flow is laminar, which lets us do some very interesting things in the lab. And then if you reduce the dimension of the channel further and you become nanochannels, then that is an entirely different physics altogether. There is something called a continuum hypothesis that we can use in all these length scale, but that continuum hypothesis actually breaks down when you are in the realm of nanofluidics. So we do not work on nanofluidics, we do not work on this sort of mini fluidics, we work in this microfluidics regime in our lab, okay? But the physics in all three cases are different and they're very interesting, okay? So now I told you there's a journal called Lab on a Chip, right? So what does it mean? It means that you take a lab like this, where you have individuals working manually, pipetting solutions from one test tube to another, or one Eppendorf tube to another. And then there are many instruments where you do some analysis in one instrument, then you manually carry that tube to the next one to do the next bit and all of that, right? So the whole idea was whatever you do in this room can be actually miniaturized and put it into a single chip. Okay. So what is this chip? This has, for example, a bunch of wells, which are reservoirs. Then there are a lot of channels that connect them. In between, to do flow control, you will have some valves and so on. There might be some other ways to do electrical measurements or optical measurements. So this is going to be something very complex, so that you can actually automate and miniaturize this entire process. Okay. So I've been told that this the background amongst you is very different today. You have biologists as well as engineers, and but all of you would have probably heard of PCR because of COVID, right? I mean, you've all heard of RT-PCR. Now, in that is slightly different, but essentially what PCR involves, you take DNA that you have. In this case, it's not DNA that you start with. You start with the RNA, that's nucleic acid of this SARS-CoV-2, the virus, and then you first convert it to DNA, and then you make many copies of this so that it can be detected by the test, right? So that process is called DNA amplification. And typically, in this DNA amplification, after that, you have to detect it in some way. Today, when someone does an RT-PCR test, it's detected by fluorescence. But in most labs, the way that amplified DNA is detected still is that they do another process called electrophoresis. We don't need to understand right now what those processes are. But the point that I'm trying to make here is that you need, first of all, these two fairly big instruments. So here you can see the length scale. This is about 1 meter long, and this is about 30 centimeters also. So these two instruments that put together, there was this paper, and this one came sometime in the late 90s, I believe. Everything that you do over here was miniaturized into this same chip. Okay, so this is the actual concept of a lab on a chip. Now, it's true that many of these lab on a chip products are not yet in the market, and there are some reasons for that. Because if the device becomes complicated, then you also need pumps, etc., a lot of other things to make it there. But it's not that it's not entirely there. There are very simple versions of these chips that are still there, and it's being widely used in our diagnostic industry. You'll be surprised to know. Okay, so let's move on. So we use microfluidics in our common day-to-day use, and we don't really know. The most common use, it's not the most, but one of the most common uses is this pregnancy test that you have. There is this kind of sort of a nitrocellulose strip. It has actually multiple materials which are porous, and fluid flows through these pores of these material, and these connected pores act like a channel. Okay, so this is, in some sense, this field of fluidics can also be used here. Another is our inkjet printer. I mean, almost many of these offices will have these printers where you can have a very tiny nozzle which looks like this, and you put drops of liquid onto your paper. So these drops are picoliter drops, and the principle is exactly the same. This is indeed a microfluidic channel that you have here. Okay, so there are this field not just in diagnostics. This is also used in other fields as well. Okay? Now, the reason I am interested in microfluidics is that because the kind of research I do today, biology has become a very interdisciplinary field. There are people like us with no formal training in biology who work in this area. Okay, and the reason is that it's become a lot more quantitative in the last 20 years or so than before it used to. And there is a lot of interest in understanding how individual cells within a heterogeneous population they look. So this, what you see here, is a microscope image of cells in a petri dish. So each of these things that you see here, for those who are non-biologists in the audience, each of these tiny things are one cell. Now, they look more or less identical, but they can actually behave in different ways depending on, if there has been some genetic change within some cell which you are not able to see in this way, or depending on the neighbors of the cell that they are surrounded by, they behave differently, or maybe the mechanical property of the material they sense that on which they are on that affects. So there's a lot of reasons why the cells within a population can be heterogeneous. But right now, given the current tools, we have no way of studying this heterogeneity. Okay, so because if you sort of try to individually address them, you need to have probes of micron dimension and do this, and this is typically not done. If you add a chemical, that's going to spread, right? That's going to go everywhere. So this is where microfluidics can help, and we will soon see how this can help. Okay. So, for example, if this is the picture of a microfluidic device, this A, B, and C are sort of three places of this device. Okay, um, it is a microscope image again. I mean, there is a scale bar. Unfortunately, I don't know what that is right now. What you see here, these objects, these are tiny obstacles, this kind of funny shapes that you have actually made inside these microchannels. They are also in dimensions of microns. Okay. And then on top of these, you see there are these small circles that are there. Those are actually individual cells. So if you have some kind of obstacles patterned in the microchannel and then you flow those cells, these geometry of these things, they actually make that cells get trapped in this. And the size of these things are such that only one cell gets trapped into one of these things. And this way, you actually, instead of this haphazard distribution, you now can have a very nice array of cells organized this way, okay? And that actually helps people to understand each of them. So now you can sort of, it's like a matrix, right? There's a row and a column number, and you can kind of see which cell you are studying, and you can go back and again watch this. So you can actually keep track of what's going on. Life becomes a lot easier, okay? But there is some inherent reason. This is the technology, but this technology comes from some science, right? And that is because of the length scales of different biological objects. Okay, so this cartoon that shows you different objects, right? So the atoms are the smallest one, which have the dimension about zero point 0.1 nanometer. Then you have larger molecules that have about 1 to 10 nanometer is sort of the size that you get. Okay, and then you have these proteins and then other bacteria, etc., which sort of proteins are still within some 10 to 100 nanometers. But as you move, this is a virus, which is 100 nanometer. Bacteria is 1 to 10 micron. And then you have what are called plant and animal cells. Okay. So with cells within our body, for example, our blood cells, right? So these are of the order of 10 to hundreds of microns. Okay, the nerve cells are very long, whereas the blood cells are typically in this 10 micron, 20 micron size. And bacterial cells are a bit smaller, they're about a couple of microns in size. So if you see this entire micron sized objects, which you can actually see with the light microscope, they match with the dimensions of microfluidic channels. Okay, and that is the reason why microfluidics is very good for studying single cells. But what happens if you want to study much, much smaller objects? So, for example, objects like these lipids or proteins and so on, you have to make channels in nanometer dimensions, which is not something we work with. Okay, there are people who focus on what are called these nanofluidics, but not our group. So, we kind of limit ourselves in this length scale. So I told you that there is something special about fluid flow in microchannels. Okay, so this is basically what is called laminar flow. So if you plot the velocity vectors at each of these points, you can see that they are sort of parallel to each other. They don't cross. Okay, they don't mix up. They are sort of like this. And as I said, as a result, if you flow two liquids side by side, they will continue to flow without mixing. And that gives you a very interesting property that if you have a cell in this region and if you have a cell in this region from the same population, let's say you treat one with the red chemical, you treat another with the blue chemical simultaneously, and you can see what they do. Okay, I will see if I can go back to this pointer and I can play this video. So what it is here, what you are seeing is the top view of a microchannel. So I'm looking at it from the top. This is the inlet, the right side. This is where the liquids are going, and there are different outlets. The way the pumping is done, that when it's pumped from there, only one of the channels get filled, not the others. And the way you do that, you essentially put those at a higher pressure, right, compared to this, so that the fluid won't flow in this way. But if this is at a lower pressure compared to this, fluid will flow this way. This is a simple pressure driven flow, right? Now let's see what happens. So first, there is this green liquid which goes here. It just goes a little bit over here, but not so much. Then you flow red. This is the area where we are interested in, okay? And then there is yellow. And this video is not really speeded up. You are watching things in real time. And you can see that you can see the different colors, blue, again some greenish thing, and red, and so on, right? So until all of it gets filled up, they'll continue to pump something or the other. So you can definitely see how these liquids are actually, you can still see these bands, right? That is the beauty of microfluidics, that you can actually see this laminar flow. And that is what gives us a lot of interesting things to do. Okay, so let me again go back to the laser pointer and tell you. So I'll before I go into our projects, I'll just give you one other example that's been published somewhere. So you can have what is called spatial and temporal control over a cell's environment. What does it mean? That if you look at, for example, let's not read this for now. Let's look at this picture. This is the picture of what is called a single mammalian cell. Okay, so all these cells, most cells actually, they have something called a nucleus. Okay, this is your high school biology, and this nucleus is stained with the color blue so that you can see them. Okay, spatial control means at least I can choose to put something on the left side of the cell and something different on the right side of the single cell. This gives me some spatial control, right? Which is not possible if you're doing this in the traditional way people do biology. And temporal control means at one time I'm doing this, but at the next instant, if I choose, I can just switch the two pumps and I can actually switch the two chemicals that way. Okay, so this is over time, you can also control things. This is a very important thing that is not possible by any other technology. So there are some organelles within the cell. So with a cell is a very complex system, right? It's filled with different kinds of things, right? And one of them is called mitochondria. You may have heard this in your high school. We used to call it the powerhouse of the cell. So the mitochondria can be labeled, or you can sort of add some dye to see it. So what they did, they flew laminar flow, right? So they flew one liquid, which is red from this side, and green liquid from this side, so that the mitochondria on the left side were stained red and the one on the right side was stained green. And then what happened? They waited and watched. What they found was how they were moving against each, like whether the green ones were going to the left and the red ones were going there, because that gave us some idea of to watch this mitochondria and how they distribute themselves in the cell. Okay, these sort of things were not possible before. Okay, so this is what was possible by microfluidics. So this is one of the very important examples of what microfluidics can do. Now, microfluidics, as I've just told you, has a lot of use in diagnostics, and there are multiple reasons for that. So, the chips that you currently see in result that are very user friendly, there are few steps. So, a lot of people with minimal clinical training can use. It's portable, so there can be point of care use. You can take it from your lab to the actual hospital or any other place where you need this. Ideally, there should be little external equipment. If you do many tests, then you can have a high throughput. It's there. You can read out in some way. So for that, you need to build in something else, whether you're reading the signal by some fluorescent dye or you're measuring an electrical current or something. There can be many other readouts. So it allows you to quantitate your readings. And finally, it is supposed to be low cost, right? Because you're using, there are biology reagents are very expensive. We are using this small scale and easy disposal. So for example, for drug delivery, etc., this is not something I work with. Microfluidics has a lot of use there. There is when you are testing different drug combinations on cells, you can do some kind of combinatorial chemistry, which you can again uh, do using microfluidics. Okay, so in the whole pharmaceutical industry, you can actually make things a lot more streamlined using this technology. So, okay, so that was the introduction to the field, and what I'm going to do now is I'm going to in about in the time that I have, I'm going to talk about two of our projects. Okay, so the first one is we sort of enriched or rather separated platelets from whole blood using a microfluidic cell sorting device, right? So let me start giving you a quick recap of what we know about blood cells. So our blood has two parts. One is the cell part of thing, and the other is the liquid part that we call the plasma, right? And there are three kinds of blood cells we know. There are the red blood cells, there are the white blood cells, and there are something called platelets. Okay, platelets are important because they help in clotting of blood. If the platelets didn't work, then we would just die of bleeding, even if we get a small cut. And unfortunately, there are certain diseases where blood doesn't cause clot well as well. Okay, so this is a problem. Now, the reason I'm bringing this up is we are now not looking at the biological properties of the cells, but we are looking at their physical properties. Okay. So, one of them is the size, their stiffness, and such things. Now, among these three kinds of blood cells, you have WBCs, which are the fewest in number. So, they have only about 5 to 10,000 per microliter, that many WBCs, which is not a lot. Okay. And then you have the red blood cells. And the WBCs, by the way, they vary widely in size. So they can be anywhere between 8 to 30 microns. Okay. Then you have the red blood cells, which are about 7 micron in diameter. They are biconcave. So there are two numbers that you at least need to describe such a shape, right? So you have the diameter if you're looking from the top, and there's the thickness, which is about 2 micron. The red blood cells are the maximum in number within a blood, with the blood sample. So you have about 4 to 6 millions of red blood cells per microliter of blood. And the red blood cell does a very important job. They carry oxygen throughout your body. Okay, so that's very important. Okay, there is something called hemoglobin within the red blood cells, which helps you do that. And finally, we have platelets, which are about 1 to 3 microns in size. And their number is intermediate between white and the red blood cells. Okay, so they're about 150,000 to 450,000 of platelets per microliter. Okay, these numbers are important because when you want to separate something that is like platelets, then you have to take into account how abundant they are, how what their sizes are, and the third thing is how stiff they are. Now, among these three kinds of cells, the red blood cells are the most soft. The reason for that is our blood vessels, as we go from the heart starts pumping blood, and you have the widest blood vessels over there, that are the largest blood vessels, the arteries and veins that start at the heart or that go to the heart, right? And then they go into different parts of the body and they kind of become thinner and thinner. They become thin as what are called capillaries. And there, these capillaries are sometimes even smaller than the size of these red blood cells. So the blood cell has to pass through this to reach the extremity of the body, and it needs to be deformable. Otherwise, your body will not get oxygen. Okay, so that's why they are deformable. But on the other hand, if you measure the stiffness of the white blood cell and the platelets, you will see that they are stiffer. So you may ask, how do you measure this? So there are many measurements that people do that measure the stiffness of single cells. So you may have heard of something called AFM, or the atomic force microscope, that gives you the Young's modulus. There are techniques that are called micropipette aspiration that give you something like the shear modulus. There are many elastic constants, right? And you can measure those. You can even use microfluidics to measure, but today I'm not going to go into that. So we now remember what we learned in high school about blood cells, okay? So what a cell sorter does is it basically, in the inlet of it, it puts, say, a mixture of cells. So there is, let's say you have WBCs and white blood cells here, okay? The platelets aren't shown in this. So let's say there are two kinds of cells that are there that you put in the inlet. Something that this cell sorter will do, as a result, you will get these two fractions separated in the outlet. That's the job of a cell sorter. Now, why we call it a passive cell sorting? Because the only forces that you have here is the hydrodynamic forces. You are not going to have an electric field or magnetic field. You can have that, but then those become what are called active cell sorters. We don't use them because they are actually pretty tricky to use. They have their own advantages, but over here I'm going to talk about a passive cell sorter device. So what a passive cell sorter device does, for example, you have something like a membrane, okay? It's like a filter. So like your coffee filter, it has certain pores, and coffee, the grain of the coffee gets stuck, but the liquid goes through that, right? So similarly here, these filters, they have different sizes. Depending on the sizes, the small green cells can go through it, and the large red cells, for example, they are stuck. The problem with this is, if you operate such a device for a long time, then these filters get clogged by these red cells getting stuck everywhere, and they stop working. So this is one typical picture of a microfabricated filter. Here you can see the scale bar is, it's 5 microns. So this is maybe about 10 micron size a hole, right? And there is one cell which has just gone into it and it's not able to go through. But that means after a point, if this happens, then the device gets clogged and stops operating. This is a big problem when you are handling blood cells, okay? So now what people typically, just what I showed you, the general principle of this is what is called size-based separation. So what it does, these are sort of cartoons where you have a bunch of pillars within your microchannel. But the gap between these pillars, that keeps changing as you move from row to row. So initially the gaps are just large enough so that your largest cells with, say, size R1, they get stopped. Next level, the things are, so but R2 and R3 can go through, right? Next level, you have such size gaps between pillars such that the cells with size R2 are stopped. And then the cells that you have is R3 are there, right? And this way, if you put an outlet here, you kind of get mostly R1 cells, but some R2 and R3 cells, right? If you get an outlet here, if you make one, then you get mostly R2 cells, but also some R3. And over here, if you make an outlet, you will only get a pure population of R3. Of course, when I say pure, ideally it should be 100%, but it's not often like that. But you can, to some extent, separate them. Just because it's not 100%, that's why I'm using the term called enrichment, as in the fraction of the total number of cells that is R3 is higher here than the fraction that would be here. And the reason simply is there are more number of cells which are R1 and R2, right? Now here, almost all the cells that are there, that are R3. So, as I said, remember this, WBCs are larger and stiffer than red blood cells. And the second thing is that if you wanted to use this scheme to separate platelets from the rest of the blood, you would come up with one difficulty. That is, the red blood cell, one dimension is 2 micron. Don't forget that. And secondly, they are very soft. So a gap that is actually only designed for platelets, even red blood cells will go through. That is the big problem in size-based separation. Okay, so we came up with something that we call the radial pillar device, or RAPID. That's short name for sorting. Um, Now, before that, let me tell you a couple of common geometries that people use. So this is called what is called a dead end pillar device. What it means that your flow is along this direction and the pillars are just across the flow. Okay, and this is the device that actually gets clogged very easily. Now, this device, it is called a cross flow geometry, where the flow is parallel to the lines of the pillars. Here, you don't have much clogging. But the problem here is that even if you have continuous operation, most of these cells, they remain within the channel. If you see the green cells, the ones that you are actually trying to separate, they are supposed to come out through these gaps on the left and on the right, but most of them, they remain here. So your selectivity in sorting goes away. Each device has its own plus point, which is written in green, and it has its negative point, which is written in red. So what my PhD student Ninad did, he actually took these two and he came up with this device, which he called the radial pillar device. So essentially, if you take a cross flow device and if you bend it and if you put it together like this, then it becomes rapid. But there is more to it. But that is the first basic principle. Okay. So here the pillars are arranged in a circle. You have one inlet in the center and you pump them in a radial direction. Okay. But you will see that what happens by doing this, we can actually get the cells out. So we can capture platelets, which is the smallest of in size, and we don't have clogging, as I'll show you a video where we run this for 6 hours with whole blood and it doesn't clog. Most devices that passive sorting devices that have been reported that can run only for a few minutes. So this is important. So now let's take a detailed look. This is an actual microscope image taken. So each of those tiny things that you see are pillars. So if you see here, the pillars are arranged in multiple rows, but the rows have also kind of 3 different zones. You can visually see that, okay? So the first zone is what we call the WBC capture zone. The second zone of pillars, we call it AD zone, but don't worry about this. This is actually the zone which gets rid of mostly the RBC zones, RBCs. And finally, the last line of pillars that we call the platelet capture zone. And this is where, after this pillar, this blue area is where you will have the platelets collected. Now, in this second zone, there is another hole that is an outlet which takes out the most of the RBCs. Some platelets will, of course, go there, but our goal is to not have any RBCs come out in this blue band. Okay, so that should only have platelets. That's the goal. Okay. The way the pillars arranged here is the key thing. Okay, so they are not straight away, the pillars are there in the middle rows rather, in this zone. You can see that this is AD stands for angular displacement, and that means that here there is a successive angular displacement between 2 rows of pillars, and I'll show you how we come up with this. Okay. So this cartoon tells you what happens. The blood goes in, you are pumping in this way. There is no other pumping. But as you are pumping in this way, here the pillar gap is about 8 microns. So most of the WBCs, they're stiff. They will not be able to go through. They get stuck here. Then what you have here is this angular displacement zone. Here the pillar gap you will see suddenly very large. It's 50 microns. That is because we are not doing what is called size-based separation in this zone. We want most things to go through. And as you see, there is a successive angle. These lines are a bit curved, right? I mean, let me see if I can actually change my pointer into a pen. So you'll be able to see. So there is sort of a curve like this, right? It's not like a straight. But over here, if you look like this, these are more or less straight lines. These are not curved lines, okay? So this is the zone where you can actually have the these there's a reason why we have them at an angle, because of this which the red blood cells get out of the region. And finally, again, here the pillars are straight, they're in straight lines, they are not angled, and the platelets come out. Okay, so this is the novel thing, this angular displacement. So okay, there is something I forgot to say, that what we do is pumping in this direction, right? That is called the radial flow. But what we don't actively do, but the arrangement of the pillars in this device setup, that is called a cross flow. That is a flow that is in the transverse direction like this, okay? And we want to maximize this cross flow to avoid clogging. It's because we have this cross flow that the cells are, most of the RBCs get out of here and they don't clog the device. And I'll show you videos as proof of what we have done. So first, before we do anything, we simulate this in console. Okay. So you see the picture of the device. The top row, this color chart that you see, the blue means there is very little flow, and then this is the red means there is high flow. Okay. So if you see here, this is showing you the radial velocity. And this over here shows you the transverse velocity. Okay. So in this particular geometry, there is no cross flow outlet. There used to be a circle in the previous picture that I showed you, right? That is not there in this geometry. Okay. Secondly, that angle that I talked about, that is not there. So first, before we made that angle, we made everything in the same line, right? One pillar next to each other. And we find that if you look at the cross flow, this color map, so blue and red essentially tell you that there are flow in opposite direction, and if it's no flow, it's white. It's entirely white. There is no cross flow. So therefore, if I try operating this device, this is the simplest design, it will definitely clog. So what's the next step that we do? The next step we do is we just punch an outlet here, right? Now you can see this. And you can now see that the radial flow also gets a little disturbed over here, because you can see things that start to move here, and you see some red and blue color. That means there is some cross flow now happening. The next level, what we do, we also make these pillars at an angle. So you can see the difference between this and the difference. So here you can see how the pillars were, and here you can see how the pillars are now. There's a slight curve. And this red and blue color also has maximized. So that tells us that we can have a crossflow in the device only when the pillars are arranged in this curved manner and we have a crossflow outlet. Now the question is, how much curved should it be? What kind of, and the angular displacement is essentially the line that you draw along the radial direction versus the line that actually follows the pillars. And there's an angle between them, that is our displacement. So you have to optimize it, right? So what Ninad did was he changed this angular displacement. Um, and again, this is sort of a zoomed portion of this. You can see this sharp red colors in between, and you can try to maximize this. Okay, and along this line, this green line, he actually plots the cross flow velocity to see what this is. And we see if as we change the angular velocity, even it's zero, which means there is no curvature, this thing is almost zero. But as we keep increasing it, then this angular displacement, the cross flow velocity also goes up. And he did that for 3 flow rates to show that, okay, whatever flow rate you use, is the same thing happens. Okay. Now, what happens after 5 is that, again, this kind of, there is a periodic condition. So, right. So this curve will actually come down like this after 5. So it's like the same situation. So it's symmetrical. Okay. So this is where you have the maximum displacement, and therefore this is the geometry that we use. Okay. Then the question is, then I'm going to show you 3 examples of this device that says that, first question is, before I even load blood, let me load some multi-sized particles. These are polystyrene particles which are stiff. So this will be purely size-based separation because they are not soft. So there is no deformability one. So whether we will have cross flow and radial flow, and whether we will get what we are looking to get. Okay. So this is the device picture, or you might say the sort of a cartoon of the device that you can see here. So this is what we call the cross flow outlet or RBC outlet. This is radial flow outlet or the platelet outlet, right? And we choose, there are 3 sizes of beads, 10 micron, 7 micron, and 2 micron, to mimic different cells, and they are mixed in the ratio. So where the platelets are much smaller compared to the rest. So, this is what we have, right? So This is actually a movie, and I have to again go back to this other pointer option for you to show. So this is a microscope image. In there you can see this is the zone 2 pillars, which is this zone, right? And this is the zone 3 pillars where the gap is much smaller, so you don't see that very closely, which is this zone. Okay, we are not really interested near the inlet, so everything that you will see will happen between zone 2 and zone 3. If everything goes well, the large particles will go like the large particles will go like this, and they will get stopped over here. So you will see this. Okay, so let's see what happens. Initially, that line just shows the liquid gets filled with the chip. And as you can see, there are these black things that are collecting here, which are essentially the particle. Okay. This is a bubble that is disappearing. And as you can see, there is a cross flow that kind of goes there. And we will see the large particle goes this way. And this is the smaller particle getting collected. Okay, so this is a picture. If you sort of zoom in even more, and you see now, and you can see in the zone 1, the 10 micron beads are getting stuck, as we would expect. 7 micron beads are going here in the cross flow direction between zone 2 and zone 3. And after that, we will collect the only 2 micron beads outside of zone 3, which is not shown in this picture. Okay. So then what we did, you have to quantify, right, the results. So what you do is you count what beads numbers you put in at the inlet, and you then count what you get out of this outlet, and you count what you get out of that outlet. Okay. And then what you find out is the throughput. That is, if you increase the flow rate, how what is going to be the number of cells coming out. Okay, so you can see that the flow as you go on increasing the flow rate, the throughput of the device also increases. That means how quickly can you do this. Okay, so this red thing tells you the number of particles in the cross flow outlet. The square black tells you what is the numbers in the radial flow outlet. That number is of course going to be kind of smaller. And then finally the blue one is the sum of these two things. Okay, so because the larger flow rate is actually through this red one, so the blue and the red one are the close to each other. Okay, this is where you see the purity. That means again, what is the purity of the smaller beads, 2 micron beads, and the 100 micron beads. And you can see and that for different flow rates, you can go something like 3 ml per minute, which is very high, and the purity always remains above 80%, right? So the population that comes out, 10 microns are coming out at the inlet, and 2 micron is coming at the outlet. And again, over here, what you have is the recovery. That is how many you put in and how many you get out. Are there any losses in that chip? And you can see the recovery is pretty good. It's above 90%, right? So, and the best thing is all of this you can do at 3 ml per minute. In a couple of minutes, you are done. So all of this actually works. Okay, so then the question is, why does this device not get clogged? And I just explained to you the theory of it. Let me just run this and show you how it actually runs. So you can see the large particle path in purple, and the blue one actually tracks the path of a small particle, and you can see that it gets out here. Okay, so this is how it happens. That is why this secondary cross flows that's come up, because you are not pumping by another pump in this direction, right? So we call it a secondary flow. I'm actually pumping in radial direction, but the geometry is ensuring that there is a second flow direction as well. And because of that, there is no stacking. Even if there are cells here, there is still, this place is open. Okay, so I have zoomed into this size and I have showed it. That's how that does not get clogged. Okay, so as I have shown you, I have already shown you this curve, so I'm not going to go through this again. So the next question was, how can we handle? So beads are one thing, blood is a whole different thing, right? Blood is soft, the sizes are not that uniform, the size ranges vary. So if I put blood in, what I would expect is at the inlet flow, the WBCs would get stuck. The cross flow would take out most of the red blood cells. And then in this radial flow, you would have the platelets out at the end. Okay, let's see if that happens. But the moment you move from this device to separate particles to a device that handles whole blood, there are some quick changes that you need to do. Okay, and one of that is you need to reduce the height of the device to 5 micron. That is because RBCs are biconcave. Okay, if you measure the size of it, you will see, and again, most textbooks would say something like 7.2 micron. That's really a wrong number. That is because the size is very, people have, these are biological objects. This is not like somebody measures them to that accuracy to do that. Okay, so there is always going to be a distribution. Okay, and you can, mostly you will see the peak is around 7 microns. That's why you call it a 7 micron size. That is the diameter, and the thickness again, of course, is about 2 microns. So what happens if I don't have this height reduced to 5 microns? So keep an eye on this green cell with this green circle and cell with this yellow circle. This green circle cell, that at moment it encounters, it turns on its side and it goes in, right? Whereas this yellow one, here the height is 5 micron. There is no room for it to turn. Let me play it again. 10 micron height means it can turn on its side because it's only about 7 micron in. If it turns, it's still 7 micron and it can go through. And this green, the blue, yellow one, by reducing the height to 5 micron, I'm forcing it to remain flat on its side. So therefore, this kind of thing will not happen. So I need to stop RBCs, and that's the first thing. Secondly, blood cells tend to settle in the tube, which doesn't happen in the beads. And the reason for that is when you are handling blood, you are actually moving things at a very slow flow rate. Okay, you can't use the kind of flow rates that you are using for your beads. So you can see this red color of settling blood. So you will have to add some sort of a vibrator motor, a tiny thing to the tube, and program it such that it periodically shakes the tube and the settled cells come in. Otherwise, your cells will not reach your microfluidic chip. Okay. And the third thing that you do is that this chip and the connectors that you put in, you need to sort of put in additional, there is this, we used an elastomer called PDMS to make it. And while the chamber height is small, we have to hold these things in place such that this is your actual chip height, right? This is the chip. This is, if you put these connectors here, this is the height. Now what happens is, if you actually keep it like this, these vibrations, etc., these things will come out. So what you do is you put it in this kind of a plastic box, and then you pour more PDMS around it to give it this, and then you remove this plastic box. So your chip, which is originally the actual usable chip, but you give it this extra material to make it strong. Chamber height, everything is the same. It's just how the solid material around it is what is more. Okay, and this is a picture of this, how it looks. So now I'm going to show you. So what you would expect is that the red blood cells would follow the cross flow and the platelets would go in. Okay. So let's see what happens. This blue thing and the red thing, if you see, the blue thing is a platelet that goes in. The red blood cell comes and stops. There is another red blood cell, because they are large. The platelets are very hard to see in general. That also goes and takes this curved path. So the red blood cells and more of those, this is a very dilute blood solution, by the way. And we did this experiment so that you can actually see individual cells. Otherwise, you would not even be able to see those. Okay? So looks like it does. But the thing is, the actual test comes when we use whole blood, not diluted. And how do we do that? So let me show it. This is a picture now with whole blood. Okay. And this is now all these red blood cells flowing this way. Some of them are getting stuck here, but platelets will continue to go through. Okay, so this is something that we actually recorded for 6 hours with blood. So these videos are there in the supplementary information of the papers that we published, and you can see them there. And this is another picture, and you can see this is the time, and this is something that is not a real-time video. This is speeded up, this one. Okay, so this is a larger view of the chip. This is the inlet. This is the cross flow zone, you'll see. And these are the pillars for platelet. These are the pillars for WBCs, and these are the angular pillars. Okay, and you see this. So this arrow that you see here, that actually is, I don't think it's actually playing. It stopped for some reason. Let me see. This arrow that you see, this blue arrow is your radial flow. And ideally, you can see, again, I don't know why I have to keep doing it, but I guess I have to just keep doing this. This red arrow is your cross flow, okay? So this is how it goes. You can see that there are some cells that are stuck, and you see soon they get released by themselves. You don't have to do anything. Okay, can you see? This is something I have now just changed the magnification, and I'm zoomed in, and I'm seeing that, how these cells go. There are loads and loads cells of whole blood. and they are still going through. There was a block here, that block got cleared on its own. Okay, so all of that. So you actually, this device can work for 6 hours without clogging. Okay, okay. Let me just again go to full screen because of. All right, so let me move to the next slide. So here again, the way we quantified for beads, we quantified for platelets, and we found that what is called the enrichment factor, that is how much you put at the inlet versus how much you get at the outlet, that ratio tells you. So the ratio has to be good enough. And we see we get something like about, I think this is more than 50-fold enrichment of the platelets as a function of flow rate. But here, the problem is that the flow rate, if it's high, then this enrichment factor is low. So ultimately, when you are handling blood, you actually have to operate this device very, very slowly, very, very slowly. That is not like what you could do with beads. That is because blood cells are deformable, especially red blood cells. Okay. The throughput, of course, goes up if you increase the flow rate, but then you lose the enrichment factor. So you have to balance between the two, and you can use something like maybe 1 or 3 microliters per minute, where you have a reasonable throughput and you also have some enrichment. Okay. If you take images, this is the inlet, you see it's full of red blood cells. If you take an image at the outlet, you will see most of these red blood cells are gone. Now, if you zoom in, these are confocal images, and you can see that these inside this blue circle are what are the platelets. This thing is a red blood cell, but these tiny things are platelets. So you can actually see the platelets that have come out of this. Okay, so this is the first part of the story. Now, before I move to my second part, I would like to know if there are any questions. If there are questions, I can take those before I move to the second part. Okay? Or we can also take them to the end. Now, this is the kind of microfluidic device that I showed you, which are of the standard materials that we use in the lab. You need a clean room to fabricate, the master for that, and then there is a lot of pumping, etc. is needed. And then there is the other kind of microfluidic devices, which is like your pregnancy test kit, which is very simple. You just use sort of a dropper to drop a sample, and it gives a result by some color change. Okay, so that is the sort of thing that I'm going to now talk about. Okay, so if you see these devices that I'm talking about, they are called what are called pipofluidic devices. By the way, Dr. Khole, how am I doing in terms of time? How much time do I have? Can you hear me?
Yes, madam.
How much time do I have?
Half
Have
an
I
hour
run out
more.
of time?
Yes,
Okay,
if
okay. Half an hour, right? I'll be done by then. Yeah, I don't want to go over time. So that's the thing I wanted to be sure. Right. Okay. So these kind of pipofluidic devices are already there. Pregnancy test is something I showed you about. Today you have these a test for dengue, malaria, HIV, and then there are things that measure various metabolites in blood. Okay, they are there. And today you have for COVID, there is this antigen test and antibody test, which are also, they work like this. These are called lateral flow assays. There is also, they are called RATs, right? This rapid test. Okay, so this is, but they all use this fluid flow concept, which is also the pores are of micron sizes. So you are essentially dealing with the same kind of fluid flow things. Okay. Now the common thing between them is the thing is made of cellulose. A paper is made of this carbohydrate called cellulose, right? Or another variation of this, which is called nitrocellulose. Okay. Secondly, here there are no pumps, no active pumps. So instead of like we have a difference in hydrostatic pressure and we drive it, here you have simple capillary action through which fluid flows. So that makes it easier. And typically there is a color change, or there are some lines that can appear that tells you that you can detect these things visually, right? So there are many such things there, but there is nothing for oral health. Okay. So when I started my lab in IIT Bombay, my first student was a master's student, and this guy was a dentist by training. So he said that why don't we do something in the area of oral health? It doesn't exist, and it's important because a lot of people, they have what are called dental caries. So there are bacteria that grow in your teeth, in your mouth, and then they kind of eat up bits of the thing. And this is an advanced version of dental caries when it's really gone bad and you can't save your teeth at all. right? And then a lot of school children we like to have sweets. So naturally, there's a chance that if we are not careful, we will develop caries. And then there's a study where we found that there were people who visited the OPDs of hospital in for various other reasons, and they were asked to give a sample of the saliva. And it was found that in Mumbai, more than 80% of people have dental caries and they do not know that they have it. Okay. So there is not enough awareness about oral health, and as such, you don't have doctors everywhere. So forget having dentists everywhere, right? So it would be nice if there's something to monitor your oral health at home. So what we developed was a fully printable paper fluidic device, which meant if you have a printer, you can use it to pattern channels, you can use it to deposit chemicals, and you can actually have a complete chip. This was nothing fancy. This was your standard, I think, some Epson printer, very old printer, about costing about 1,000 to 2,000 rupees, which we got, and we kind of hacked it such that where you load ink, instead of that, you can put tubes with chemicals, okay, and you can then print. So the whole idea was we would do one level of printing where we would, this dark gray area is where you would have your um, um, the hydrophobic material, which is something has to stop the fluid from going everywhere, right? So you need what are called hydrophobic or water repelling parts, and then there are these light gray parts, which are the parts where you want liquid to flow. Okay, so you do this patterning by loading one kind of thing. Then you put some reagents, which we do two tests, and you instead of load it, and then this pattern paper you put in the printer again, and it now prints in the regimes these kind of two chemicals, and then if you add your saliva here, you would go. Okay, so how did we pattern here? And again, there are some fancy ways to do it. The way we did, we just added some olive oil and in the solvent, which was isopropanol, so that its viscosity and surface tension kind of was in the printable range. So you know that water has very high surface tension, right? So water needs some surfactants for this to be printable through this thing. So if you look at the inks, if you go to say, we are using an Epson printer, right? So if you go back to the published patents and you see Epson's patents on the inkjet inks that they develop, you will see that it gives a certain surface tension range for which it works, right? Which is about I think it's some 20 millinewton per meter or something. So you have to bring your olive oil in that surface tension and that viscosity range for it to be printed through this. Otherwise, it will just get blocked. So that is what. So you do all of those things, and you can build this. So you can see over here, he wrote the word microfluidics, which was supposed to be hydrophilic, and the rest, this light gray part in this picture was hydrophobic. And then when we put drops of water, you can see hydrophobic part, the drops are still as drop, but in microfluidics, it has flown and made it appear grayer, and this is how you do this. Okay, so this is just to show that this can actually make things better. So let me go through the details. So this is the picture of this modified inkjet printer. So instead of where you load the cartridges, you load what are these 1.5 ml tubes. And you can even print proteins and other dyes. For example, there is one place where we printed an indicator dye in a gradient. So this is there. And then there is the protein, that is the protein is highest here and lowest here. Indicator is highest here and lowest here, and in between you can see this blue color indicator. It changes its color and it becomes print. So you can do everything with this. So you didn't need a fancy bioprinter or anything to do this. So this is where we oil printed things, and we wanted to know how hydrophobic they are, right? And to know how hydrophobic something is, you have to measure what is called the contact angle. And you can see if the contact angle is above 90 degree or so, then that's good, that's hydrophobic for you, right? So what we do is, what this guy did, he basically he wrote this device that he printed over here, right? So he prints this and he puts it in the oven for about for different from 0 minute to 24 hours. He printed many devices. After every 1 hour, he would sort of, or every 2 hours, he would take out the devices and he would measure the contact angle in them. For 24 hours, he kept on doing this experiment, and that's what resulted in this curve. And we found that at about some 8 hours or so of keeping in the oven, this contact angle of this oil printed region, it goes up to what is acceptable for us. After that, it doesn't vary so much. So if you just keep it between 8 to 12 hours in the oven, so you print them in the evening, you leave it, you go home, you come back next morning, you are ready to use it. And these are the pictures of liquid droplets. So you can see that at 4 hours, it's still spreading, which means it's not hydrophobic enough. But at 8 hours, it's hydrophobic, and 16 hours, it's very hydrophobic, right? So that's what. And then, of course, we did some kind of FTIR to know that there is this peak that comes, which tells us that, okay, there has now, paper has been modified by oil, and because of some peak at this wavelength it comes. I don't exactly remember what group it corresponds to or what bond, but there it is. So for oral health, what we do after we pattern this, we then take two reagents, okay? And then one is for pH test, which is going to be like a pH paper, but except the change is that you can't use your standard pH paper. That's because your saliva pH is kind of varies between 4 to 8, okay? And at this range, the color change in the normal pH paper is not very strong for you to tell that there is a color change. So you have to come up with a reagent for measuring pH, which will give good change in color in this. Plus, it should if it should be in a liquid form, so that that surface tension and viscosity again match, and you can print them. So we bought a bunch of liquid pH reagents as well, and we found finally a combination of bromophenol blue and bromocresol green at a certain ratio that gives you the good change where you can change color. And this is another thing where you actually print a second chemical, which what does is basically as a reductase assay, it's called. Solubility reductase assay is a surrogate measure of how much bacteria there is. If there is bacteria, they produce this enzyme reductase a lot. So if there's more of reductase, that tells you there is more than what is good for you to, that number of bacteria to have in your mouth. There are always some bacteria, always there, but there's a basic level and there are too much, right? So this tells you if you have that. So let me first talk about this Curie susceptibility assay. So what it does is there is some kind of a redox indicator called resazurin that comes. So it detects this enzyme there, okay? So there are different lactobacillus, then streptococcus mutans, and there are some other organisms that are there in the saliva. And it checks whether these are there. They'll all produce this enzyme reductase, right? Now, resazurin is normally blue in color. So when you go to a dentist clinic, if they want to do this test, they'll have this thing in a tube. They'll ask you to sort of spit inside this tube. And if there's enough of this reductase enzyme, it gets converted to a pink color like this. That tells you that's not good. If this gets, the color changes, that means you are having more bacteria in your mouth and you should sort of do something about it, okay? But instead of doing it in this tube, where people actually have to spit a lot, and when we were doing this test, we realized that it's very difficult for people, some people, to sort of just spit on demand on this like this. So whereas if you just touch your with a toothpick on your tongue and you just put it here, that kind of works. So we have these things printed on this. Then once this is printed, we sort of scan this chip using the scanner, the printer, a scanner that you have in the lab, and you do a color analysis. So what you do here is, this is where it is sort of going to be less reductase. So it's going to be blue, more reductase, it's going to be pink. So this is how the color change actually happens. We did this, a check between a tube-based assay. So we did this over here as well as we did this over here. And we measured that this is what is considered the gold standard. Therefore, does it match with this? And if you see, if you plot this, if the color change is similar, then you should get a line with the slope of somewhere 1, right? And you can see this is the fitting equation of the line. So it's 1, and there is, of course, some this thing, but that's not so important. This is the important bit. And you have a decent fit. And we did this for 72 saliva samples, okay? Then the question comes for the pH. As I told you, the pH is actually, this is the healthy range of pH in the mouth, because this is close to neutral. So if this green color then changes to something like yellow, then the problem is that that means your mouth has less pH, it's more acidic, it's not fit for your teeth, okay? And again, we generated what is called a calibration curve, where we added solutions, made solutions of different pH, and we took these pictures. So this is a picture of this calibration curve that you see. And then we saw that, and this thing, we analyzed the color, we plotted the red pixel intensity because this was the linear region, and then we saw. So the whole thing, as I said, it had to be made printable, that's where, and it had to give this distinct color change, plus it had to be linear. So we optimized with a different range of pH indicators to get this. So finally, once this is there, we wanted to test it with real samples. So we have to come up with a test, right? So what is this? So if you're look at these colors by visually and you analyze the score there, and then you add up these scores, okay? So this row at the bottom is where the score is highest, and that's when if you add both of these, it's about 7 and 7, 14. That means it's excellent. But as you go up, your score can go up because either the pH test is coming out to be bad, or your maybe this is coming out to be bad, or maybe both are bad, right? So you can have different combinations of things, right? So when you are in this regime, it says the oral health is really bad. You need to do something about it anyway. Maybe the tooth can't be saved. You have to go for an extraction or something. Then about 6 to 8, we called it, this needs monitoring, counseling. 9 to 11 is you're fine, but you need to do monthly checkup. This is something the dentist used, and because the student was a dentist, he came up with this sort of a chart. Okay. And then we went to one of the colleges in Mumbai, it was Sophia College, I remember. I gave this, we talked about this oral health some years ago. I gave this talk, and one of the professors says, we are having a dental camp in this college, maybe in about a couple of weeks. Do you want to come out, and whatever you made, do you want to have this test? So we did this test over there, and we found that there was a very good match between, I'm not going into detail of this because I don't want to take up time. I want to leave some time for questions. But essentially, we found that we tested about 17 students, and we found it was very good match with what the dentist gave and what our test came up with. Okay, so I'll stop here with this, and I would like to acknowledge this whole bunch of people. So Ninad is the person who did a lot of the cell sorting work that I showed you, has almost entirely been his, that is his PhD thesis work. And Amar is the student who's the dentist who came up with all of this. But there have been other people who contributed to different projects in the lab. And I am also thankful to a lot of collaborators and for the funding agencies. And I'll be happy to take any questions. Thank you for your attention. So let me come out of this presentation mode and go back to this so that, yeah. So are there any questions, or is everything very clear?
Participants, any question? Otherwise, you can type in a chat box.
So I'll stop sharing the screen, I think. Yeah, I think I have stopped sharing. So. Okay.
Okay, there is no question.
Okay, all right.
Arundhati? Okay.
Thank you, Garima, for your message.
Okay, ma'am. On behalf of the organizing committee, I would like to thank you, ma'am. Thank you so much.
Yeah, you're most welcome, and it has been a pleasure to come and give the talk. Thank you very much again.
Thank you so much.
So I'll now leave the meeting and hope to, if there are anything that you want to get in touch with, please do. Thanks very much. Okay, bye.